So I don't want you to get too excited. This is only about 7 slides, but there's a lot of data on each slide. I mean, it's not difficult, but there's a lot. Okay, so the first group that we're going to go through are the agammaglobulinemias, which is a tongue twister. Okay, it has, I think, probably way too many vowels in it. Nevertheless, this is what we're stuck with, and the name obviously tells you what it is. A meaning not, gamma globulin, we know what that is, right? So these are your the antibody deficiencies. Generally, these are going to be related to B cell system deficiencies, okay? Because the B cells make the gamma globulins, okay? So now this doesn't come from literally IgG. This comes more from the fact that immunoglobulins migrate to that gamma region for serum electrophoresis. So that's really the test that's probably going to note your gamma region is going to be altered to missing. Okay, it could be anywhere in between there. Okay. So there are several different mechanisms that might be in play here. You could have some genetic defects that prevent or halt B cell maturation. You could have mutations that lead to defective interactions between T cells and B cells, which you know are required. So there's several different things that could be going on here, and so we're going to discuss a few of them. Now, pardon me. There is a wide range of deficiency states based on which immunoglobulin is missing and the severity of the deficiency. So you can see already from the names, you have some that IgA deficiencies or IgG deficiencies. An IgA deficiency would be much less significant because of the species of immunoglobulin that it is. IgA is not as important in our primary immune response, in our ability to ward off infection as IgG is. And so you're going to see different clinical signs depending on how deficient is the patient and exactly which clone is missing. So it's important to remember, again, which species are we talking about, but then also during the other thing we have to remember is that when we're trying to diagnose this kind of thing, it's not like we have a very tight level of immunoglobulin throughout our lifetime. Our levels of immunoglobulin change not only with our own state of health, but also with age, okay? A pretty wide range there with age. And that actually becomes an issue with the very first thing, which is a diagnosis, but it's also a normal clinical state, okay? Babies, for example, we all know don't have a fully functioning immune system. And for that reason, they experience very low levels of immunoglobulin for about 5 to 6 months. Now that's normal. That's the transient part, right? That's normal. However, some babies take longer. Okay, so now this is where you're getting into, this is unusual. A baby who is 10 or 12 months old, they shouldn't have these very low levels of immunoglobulin, okay? So it's these babies who are experiencing more infections, and they're usually pyogenic infections, meaning fever producing. So these are, a baby who's got a snotty nose. Yes, do they have an infection? Yeah, but do we really care about it? No, not until a fever develops. Okay, well, you might if it's there first, but the moms get over that pretty quickly because you'd be at the doctor's office every day if you went for everyone. However, fever's different. We watch when there's a fever, okay? So now when the baby is just born, the mother's immunoglobulin is there. That'll have a protective feature. So you're not going to see babies days or weeks old who, have this problem. But that immunoglobulin is going to clear, which also will depend upon the health of the baby, blood flow, and all that stuff. But that is basically going to clear. You're talking a month or two, and then the baby is going to be still exposed to the normal viruses and bacteria that are in the area, and they have normal cellular immunity, right? So nothing particularly wrong with that end of the immune system, but they don't have normal, their B cells still aren't really up to the task, if that's a good way to say it, okay? And when we test them, we actually can find normal levels of other species. We might find, for patients where it's an abnormal hypogammaglobulinemia, they might have normal levels of IgA and they might have normal levels of IgM. But IgG is the most common species or clone that will be affected. It'll be dropped. Again, this is where we have to know what our normal ranges are, but it'll be dropped at least 2 standard deviations below the mean for an infant range, okay? Now, why does this happen? We actually don't know, because obviously, again, think about who would you do this testing on? By the time we know this, now you have a parent who's frantic about their child, and you have some researcher geek coming over and saying, can I study your kid? The answer is almost universally no. So this is not well studied because it's hard to study it, just, you know, logistically speaking. The patients have normal levels of B cells. That they have not found a reduction in the normal levels of B cells. They have not found any evidence of X linkage. Even though it is more common in males, they haven't found any evidence of it. So the one theory or hypothesis is that there's probably a delay in maturation of some component within the immune system. It's probably a T helper cell, because remember, we need those, right, for all of the phases of B cell function, including isotype switching. So there's probably some delay in maturation of one of those components, and but again, it hasn't been proven. That's just a hypothesis. So it's still going to be transient. These kids are going to come out of it, but unfortunately, they're going to go through some pretty, harsh infections until they can, okay? And it's going to look like they've got no immune system on that side of it. The next one we have is the X-linked Burton's, pardon me, I wish I had a cough button. Bruton's A-gamma-globulinemia. And this, from the name, you can tell it is X-linked, not surprising there, which means it will occur more in males. Okay, so, and that's because the BTK gene, or Bruton tyrosine kinase gene, which is carried on the X chromosome. So if you get a faulty copy or no copy of the BTK gene on one X chromosome, us ladies can look to our other X chromosome for the other copy, and the gentlemen are unfortunate that they don't have that copy. So what does the BTK deficiency lead to? Well, the biggest problem is that you end up with a lack, your B cells, there's really almost no mature B cells. So the marker that they're looking for, this is the CD19 marker. These, if you were to do a flow on these patients, they would have very low to almost no CD19 positive cells, and that would depend on how far off was their BTK gene on the X chromosome that they do have. So, and this means with no mature B cells, that means an across the board lack of immunoglobulins, all classes, okay? Never got any to start with, so no classes. Without mature B cells means no plasma cells in any of the lymphoid tissues. So if you do a lymph biopsy, on any one of these kids, and these are usually diagnosed in kids because of the repeated illnesses, they'll have pre-B cells in there, right? And they'll have them in their, they'll have pre-B cells in their bone marrow. But remember, the differentiation is arrested in the bone marrow. The B cells don't migrate to the lymph nodes until after they've gone through at least some phases of maturation. It's not complete maturation where they're producing immunoglobulin. They're considered immature or latent, or, there are several words you could use for it, but they still have the CD19 marker present. Without that, they won't migrate out of the bone marrow. So that actually affects the development of the lymphoid tissue. Without those B cells taking up space for all intents and purposes, these kids are going to have, they're actually going to have really small tonsils, adenoids, right? The things that just keep getting bigger and smaller when you get infections over and over again as kids. And that's because there's no germinal centers in the lymph nodes. You need T cells and B cells to make up a germinal center. With no B cells there, the T cells kind of don't know what to do with themselves, and so no germinal centers at all. Now, if we continue our analysis, our T cells are normal in these kids, and they function fine as well. So with T cells, you have pretty good protection against viral factors. You don't necessarily have the same kind of memory that you would have if you had B cells, but you have protection. You still have memory through your T cells, right? You have T cells that become specific and maintain their memory. So the thing that these kids will have without any B cells at all, they're going to have recurrent bacterial infections. The most common are still going to be upper respiratory because that's just the most common, okay? And that even with your GI tract, you do have some protection just through the production of acids as you get old enough to have eaten normal food, okay? So these kids are going to have recurrent bacterial infections. Their susceptibility to viral infections will only be increased in the sense that they don't have the same memory. So they do have slightly increased susceptibility to viral infections. So one of the things that we do avoid with these patients, since they can't produce an immune response, we do not give them, an immune memory. We do not give them live vaccines, okay? Yes, they will produce a T cell response against the live vaccine, but they won't produce a B cell response, which is what we're looking for when we give people vaccines. So we don't give them live vaccines. Giving them a killed or component vaccine is fine because you can build up some T cell experience, but you don't risk it. It won't overcome the patient's immune system. So this is again, it's a genetic disorder that ends up causing arrested development of those B cells. The cells are there, but they're all stuck in the marrow in an immature state. So what do we do for these guys? Well, there isn't a lot that we can do. We can't give you B cells without doing a bone marrow transplant, which, although there's not a lot of research with this. These are as children, they're pretty sick, but we can give you immunoglobulin, and that's what we do. We give you infusions of immunoglobulins and then aggressive antibiotic treatment of infections. Although obviously a bone marrow transplant would be something that you could potentially do, they still don't have the gene. Well, if you gave them a bone marrow transplant, you would obviously give them one with a gene. Okay, so that would make sense. Okay, next thing, the IgA deficiency. We kind of talked about this before in blood bank because it is an issue in the blood bank. This is actually the most common immune deficiency that we find as far as what we see is 1 in 500 persons of American or European descent have an IgA deficiency. Very, very common. And the reason it's, probably the reason it's so common, is that most patients are asymptomatic. Even if the patient does have symptoms of an IgA deficiency, you might see increased number of respiratory and GI tract infections. They might also have a tendency towards autoimmune diseases, but a symptomology with this is really pretty rare. For these the patients who do have symptoms, it's usually because about 20% of patients with IgA deficiency also have an IgG subclass deficiency. They usually are deficient in IgG2. So that is where you usually see your symptoms. How do we define it? Well, if you have a serum IgA of less than 5 grams per deciliter, you're considered severely deficient, okay? But anything below 2 standard deviations for your age-adjusted mean, that would obviously be considered a deficiency. It's less than 5 where we know, oh, that's a severe deficiency, okay? The biggest problem for us is the production of an anti-IgA. If you don't produce IgA or you produce it at such extremely low levels, you conceivably could produce an anti-IgA. And again, you produce that, for the most part, not a huge problem unless you receive a transfusion with a component that contains IgA in it. In that case, that would be you would actually develop a transfusion reaction, a pretty serious transfusion reaction, and in some cases that could be the first sign. In many cases, it can be the first sign that you actually do have an IgA deficiency, because you've been in the lab, especially those who've been in chemistry for a while. We don't do a lot of IgA level testing. It's not the go-to test, right? We do a lot of CBCs, but you wouldn't see that here, okay? So IgA wouldn't necessarily come up, okay? So how do we treat this? Well, really the only thing we're really going to treat on this one is to really make sure that any products that they receive from us, any components that they receive from the laboratory, are washed to remove that IgA, okay? I mean, you can collect it from an IgA deficient person, and that would be fine, but the most common practice is really to wash it. There are IgA replacement. There has been research as far as IgA replacement, but the half-life of IgA is really, really short. It's only about a week. And if I give you IgA in your arm, well, that's not where IgA works. So it hasn't really demonstrated any, useful results. So IgA replacement therapy is kind of not really thought to be useful. Okay, the next one is a common variable immunodeficiency. This is actually not just one disorder. Common variable is that it's actually a group of disorders, and the whole group has a prevalence of about 1 in 25,000. Okay, that's pretty rare, but it is still the most common immune deficiency that has a severe clinical presentation. Okay, so in the cases where we've talked about them before, they're really not considered, well, obviously IgA is not considered severe, but X-links, they're not really considered severe. Common variable immunodeficiency is considered a severe clinical presentation. This is one that, unlike the others, you probably won't see this. This doesn't have an onset of infancy. The average onset for this particular group of disorders is in your 20s and your 30s. So you guys are pretty much almost out of the woods. And what happens is the common things that we see with these patients is that they develop things like shingles at a much higher incidence than normal young adults. And when you start testing them, you start running your tests and seeing what their levels of immunoglobulin are, is we find that the patient usually has a deficiency of both IgA and IgG altogether. And it is possible that the IgG can be a selective deficiency, but usually you're going to see both the IgA deficiency and the IgG deficiency together. And what you end up with is, it's called a sprue-like syndrome. So this is well, it was called sprue. Now it's called celiac, okay? This is it's a malabsorption. It's a diarrhea kind of disease where the patient really isn't getting the same kind of nutrition that they need out of their food, and they're also experiencing a lot of GI symptoms. It's also associated with an increased risk of several of more than one lymphoproliferative disorder, including things that could lead to gastric carcinoma, as well as autoimmune problems. So you end up with sort of a group of clinical pictures, more than just one, that will eventually, these do take time to come to a conclusion with, but what happens to the patient in general is they end up with these recurrent infections. And when you're 20 or 30, you usually think you're past your days of constant GI issues. So they're getting these GI issues at an increased rate. And so testing for immunoglobulin levels might be normal. And so they demonstrate reduced IgG. That's the most common one that that we see first. And we look at IgM, we also probably will see a reduction in IgM. They can be low to absent, okay? Now, when we do their cellular tests, they do actually have normal numbers of B cells, but those B cells aren't differentiating into functioning plasma cells. And there have been 3 different defects that have been associated with these pathways. And they can have any one of the 3. It would be really bad if they had more than one of the 3. But basically what we see here are a situation where the T cells, or the products they produce, are suppressing the B cells at a higher rate. So they suppress B cell differentiation too quickly or too strongly. The other defect is that T cells are failing to provide adequate stimulation for the B cell differentiation. So one of the things that's required, if you remember, for B cell differentiation is that T cell link between the B cell has to be pretty strong. And if it's a weak linkage, the B cell is not going to differentiate. So that's one of the issues. Usually that weak linkage is because the specificity for the antigen between the T cell and the B cell is not the same. But in this case, the reason for the weak linkage is actually because of a flaw between the connection between the T and the B cell molecules. So you have your markers that they normally interact together. And then, of course, they could always have a primary defect in the B cell line. So you can see that 2 out of the 3 reasons for B cell failure are actually T cell failures. Okay. So this patient, these patients, it can be, it's usually more than one class. It's usually A and G. You're going to see a reduction in M overall, however. So many different combinations of reduced clones in the when you do your immunoglobulin electrophoresis. And so we'll treat them by replenishing immunoglobulin to them. This isn't a case where we're just trying to replace IgA. In this case, we're trying to replace all the clones, in particular IgG. They'll still probably retain a pretty low IgA at the mucosal surface, which would put them at increased risk for respiratory and GI infections. Okay, so in that case, there's not much we can do necessarily to completely eliminate the symptoms. And the diseases that they can experience because they lack all the clones can be pretty severe. Then we have isolated IgG subclass deficiencies. In any case where you have the level of any one of the subclasses being 2 standard deviation lower than the age-adjusted mean, you have a deficiency by definition, okay? We know what that means. And I've given you here the relative percentages of which classes are deficient. And you can see, I'm sorry, that these are these are the relative levels that should be there, right? You should have mostly IgG1, mostly, then 2, then 3, then 4. Depending on which class you have, it might not be immediately obvious that in the IgG total, which would be the first test that you would do, that you have a problem with your IgG level. So in other words, we don't immediately start out our testing by doing subclass relative quantities, right? We don't do electrophoresis right away. We just look for the total IgG. That's our normal thing. Well, if your first deficiency is in the IgG3 level, and even if that's completely missing, are you really going to notice a 6% difference in the total IgG? Probably not. So really only if it's 1 or maybe 2 would you note, depending on how severe the deficiency is, would you notice in the total IgG that first deficiency that you develop. So the patient will be experiencing more and more recurrent infections. And if that's the case, if these infections are recurrent, then subclasses should be ordered. Total IgG is not sufficient. If the patient is having recurrent infection, then the physician really needs to be educated that really it is probably the subclasses that they should be ordering so that you can see what's going on. Now, the subclasses are moderately specific. If you remember, the classes 1 and 3 are generally directed against protein antigens, whereas classes 2 and 4 are generally directed against carbohydrate antigens. Now, that's not a hard and fast rule. Again, it's generalization. But we do see some correlation. So if you have IgG 1 and 3 deficiencies, you'll probably see an increased susceptibility to protein antigens, like things like toxins. Okay, whereas IgG2, when you're talking about carbohydrate antigens, like as in 2 and 4, 4 is really low, but anyway, with 2, you're talking about carbohydrate antigens. Those are the antigens that are found like on capsules of bacteria. And so we frequently, if that's the immunoglobulin that's missing, we see increased bacterial infections from organisms that have capsules. Organisms like Haemophilus influenza, Neisseria has a capsule, Staph aureus can produce a capsule. So capsule producing bacteria. Okay. So what leads to the subclass deficiency? Well, a lot of them have things to do with transcription defects. Remember, isotype switching is pretty delicate balance, making sure that you move to the right isotype. And if the heavy chain is deleted, obviously that's going to be a deficiency overall, right? So if the heavy chain gets deleted from a class of cells or from a precursor cell, God forbid, there you go. That's the whole thing. You delete mu, you're not getting IgN at all. But with the subclasses, you have, especially with IgG, you have those 4 sections after it. So it's usually the IgG1 portion of the gene, it's a smaller portion, that'll be deleted, okay, if that's the one you're missing. Now, IgG4, they put a little caveat here. It is actually the most common deficiency. We frequently see patients, frequently, these are still rare, right? But of the IgG subclass deficiencies, the most common one is the IgG4 deficiency. And there's likely a reason for that, and that is it has almost no clinical significance. Why? Because it's only 4% of the total IgG. It has very little function, day-to-day function in your immune health. IgG1 is the least common. That's the least common one to be missing. And then the last one that we're going to go through is the immunodeficiency with hyperimmunoglobulin M. Okay, so it sounds weird, pardon me, to have deficiency and hyper in the same name, but there you go. It's called this long way immunodeficiency with hyperimmunoglobulin M or just hyperimmunoglobulin M syndrome. Okay, in this case, the real problem is isotype switching. So in this case, the patient can actually mount an immune response. They have functioning B cells, but there is a CD40 antigen defect, and that, pardon me, it doesn't allow you to switch from IgM, which we all know is the primary antibody response, switch from IgM to any of the other isotypes. So the B cell will produce IgM, and it'll never get to any of the other ones. Because it's producing IgM, first of all, it's only producing IgM, you're going to have a higher level than normal. But it keeps trying to switch. So it keeps churning out that M until it can switch, and it never does. So it just keeps trying, spinning its wheels. And the reason, there are several reasons several genetic reasons why this can happen. There are both sex-linked and autosomal reasons that can lead to, it's basically a deficiency in CD40 on T cells. You don't have CD40 on the T cells, it will not bind to the B cell, causing the isotype switch. So, and if we have deficiencies in the B cell line, also, we can get deficiencies in cellular immunity. Makes sense, right? So you can see there are just more, if you think about it, B cells are B cells, right? They do the same thing, but there are lots of different types of T cells. That's why it's a longer list, okay? And these immune deficiencies are certainly much more difficult to treat. First of all, because they don't produce a soluble product that we can infuse in them, right? We can't collect the immunoglobulins out of me and put it in somebody who's missing them. So there are, they have tried cellular replacement therapy. Unfortunately, T cells, what's the problem? Graft versus host. So if you would risk graft versus host disease, or they're foreign. They're going to be destroyed by whatever immune system that the patient does have. So they usually don't, it's not kind of a safe mechanism to go around. These are considered okay, let me just get to the area where I want. For these patients, irradiation is really important, and that's because they do have some T cells usually. The T cells that they do have, however, don't function properly, and so these patients are at a much higher risk for graft versus host disease, even in something with a very small number of T cells, because their own T cells are unlikely to be able to overwhelm any foreign T cells that make it into their system. So if that were to happen, the T cells, even from say like a component of packed red cells, would be a problem for these patients. So radiation is really important for these patients. So if we go down these one by one, we have the DeGeorge anomaly. This is actually a pretty easy one to remember. It occurs during embryonic development. And this is, there's actually, during development, I remember if you took embryology, but you went through different periods of time, and you produced these different, you had gill slits at one point and all that crazy stuff. But as your neck, I guess, was developing, an anomaly occurs between the third and the fourth pharyngeal pouch that ends up affecting thymic development. If we look genetically at these patients, they're usually missing. Most patients, not all, but most patients are missing. They have a region of the 22nd chromosome that's missing. Now, that's not required for diagnosis because, again, most but not all patients demonstrate it. But without the thymus and any other tissues that would arise from that area, you're going to get, and this is a genetic disorder anyway, you're going to get some serious effects later on in life, or after birth, I should say. There is, as with many genetic deletions that occur, there is a definite mental delay that occurs for these patients. There's also some serious problems around the neck and chest. You have an absence of ossification of the hyoid bone, so it never completely matures. You have cardiac anomalies. And as with many other genetic disorders as well, they have an abnormal facial development that is common to the syndrome. Of course, they'll also have thymic hypoplasia, so a very small thymus. The severity and the extent of all of these symptoms is variable. You can get very severe thymic hypoplasia, for example, or you could get, mildly severe. And a lot of that depends on how far off that anomaly was from normal during the developmental phase of embryonic development. So the most severely affected kids will present actually in the neonatal period. The patient will actually, the fetus will actually have neonatal seizures, which is called tetany. And that's usually caused by problems with their parathyroid gland. They don't produce, they're not able to manage their calcium, and they end up with seizure and cardiac defects, but they're occurring in utero in some cases. Okay. So those would be the most severely affected of this patient group. Once you are born and we can do a diff and things like that, we do find that there are some T cells there, but they're generally reduced. They'll function if they're there, but they're definitely reduced. And so these patients will have all the problems that go along with reduced T cell immunity. They'll have recurrent viral, recurrent fungal infections. That's your T cells are directed usually primarily against those. And it is possible now, not sure where, but it is possible if you can diagnose this early enough that you can do a thymus transplant. That's a fetal transplant, however. They also have seen some success with bone marrow transplants. Barring either one of those situations, you have to treat the patient with thymic hormones. You have to give them the hormones that the thymus would produce. Second on our list is the purine nucleoside phosphorylase deficiency. This is a relatively rare autosomal recessive trait. It presents in infancy, and the patient will present with recurrent pulmonary, and it can be recurrent or chronic. I want to point that out. Sometimes it's not necessarily happening over and over again. It's just the patient's never getting over it. Okay, so recurrent or chronic pulmonary infections, and that's anywhere from like oral all the way down to pneumonia, anywhere in between. Then they also have GI symptoms, skin infections, UTIs, and failure to thrive, which I'm pretty sure you know the definition of now. If the child's not growing, they're not gaining weight. Okay, they're not hitting their milestones as infants. This PNP, or which is much easier to say, is an enzyme that's involved in purine metabolism. And so not having that particular enzyme ends up, leaves the patient with a moderate to severe defect in all cell mediated immunity. And even their humoral immunity, it can be normal, but it probably will be at least mildly impaired. So what happens then, since purine is not completely processed by the cells, you end up with purine metabolites, a toxic one called deoxyguanosine triphosphate, building up in the T cells. So what starts out as looking like pretty normal T cell population will decrease from the beginning of the disease. Your Ig levels, they can be normal and they can actually be increased. Remember, you have hyperactivity to compensate for the reduced activity on the other side. So IgG, your immunoglobulin levels will be normal to increased. And the patient is going to experience neurological disorders to such an extent. Remember, this is where your T cells are decreasing over time. So in some sense, it actually resembles HIV. So these patients are frequently misdiagnosed with neonatal HIV because their T cells decrease over time. And so they frequently are started on therapies for HIV when that's not what the problem is at all. How could we differentiate it from HIV? Well, do an antibody test. It's not that hard. Look at, use anti-HIV antibody, and it's not that hard to differentiate it. So we can't forget that there are T cell deficiencies that are separate from viral infection. We can also assay for PNP. I mean, that's not a regular test. That's a special test, but it is there. So PNP is very a specific genetic deficiency of this particular enzyme. Then we come to SCID, or severe combined immunodeficiencies. This is a group of disorders that share the same name, and it is considered the most serious of all the congenital immune deficiencies. These are a group of disorders that will affect both T and B cell function, but for different reasons. Okay, so depending on what form of SCID you have, you might have a very severe disease, which is where it gets its moniker, the most severe of all of them, to other forms of it that are a little bit less severe. Okay, more treatable, in other words. The most common form is the X-linked form. 46% of all the cases of SCID are X-linked, so that means they occur more frequently in males. And that particular form, remember, it's the most common form of SCID, but SCID still is very rare. So this particular form occurs in 1 in 50,000 births in the United States. Okay, and for this X-linked form, the problem that you have is an abnormal gene, or a gene that's missing for that is common to the gamma chain, and that's the first one listed here is the IL2RG gene is either abnormal or missing, and that gamma chain unfortunately is used in the production of several cytokines. It's used in the production of interleukin 2, 4, 7, 9, 15, and 21. It's a long list. And so there, you can see what the problem will be. Those are massively important to total immune function. Okay, and that IL2RG gene is found on the X chromosome, hence the linkage. So for these patients, your normal cell signaling that's required for a healthy immune response can't occur. So cell maturation stops in many different cell types. And depending on other defects that may be present, you can have even more severe disease, which doesn't sound like you'd want it, but it does happen, unfortunately. And another defect that could be present is the JAK3 defect, and that JAK3 is a signal transduction molecule, and it's required for processing any interleukin binding signal from the cell membrane to the nucleus. So that's your nuclear signal transduction, right? We know what that is. So the JAK3 binds the interleukin and takes that signal into the nucleus, that mechanism. So if you have a defect there, now you've just amplified the problem with that interleukin issue, okay? Now you can have just the JAK3 alone and you already have a problem, or you can have them together. And JAK3, if it's present in a B cell, it leads to antibody production. If it's present and proliferation. If it's present in a T cell, it'll lead to proliferation. So you need that. You'll see where maturation is just going to be halted. You're not going to get a complete immune response in these patients. Generally speaking, the JAK3 is found along with the IL-2RG. So that's the X-linked form of the disease. However, you can have JAK3 alone, standalone, which would be an autosomal. JAK3 is an autosomal gene. It'll be an autosomal form of SCID. It is recessive, so hopefully you have another good version of JAK3. Basically, that entire list of interleukin, the 2, the 4, and so on, these cells can't transmit any signals from those cytokines. And so these patients end up with massive both T and B cell deficiencies going on, okay? Another issue for SCID patients, and this is a lower percentage of our patients, only about 15 to 20% of our SCID patients have this deficiency, which is an adenosine deaminase deficiency. This is an autosomal gene. ADA is an autosomal gene. It's found on chromosome 1. And adenosine deaminase is part of the purine metabolism pathway. So what happens then, if you don't have adenosine deaminase, you end up with toxic purine metabolites accumulating in the lymphoid cells, and that's going to impair proliferation of both the T and B cells. So your T cells are going to build up these toxic metabolites, and then the T cells become dysfunctional. Not being able to function means that both, obviously they're not functioning, but without your T cells, your B cells aren't functioning either. Okay, and so that's why it's combined. The ADA is a much more progressive disorder. The patient develops, their T cells are squandered. They're not squandered. That's not the right word. They're progressively falling off. So the numbers are going to reduce over time. And now the patient might have an ADA gene, but it might be, if it's a complete deletion, it'll be a severe reduction. If it's a partial change in the gene, depending on how bad that particular gene is being translated, it'll correspond to the severity of the disease. Okay. Then we have another unfortunate dysfunction that occurs. In some infants, we find that the patient has neither T nor B cells, but they do have NK cells. The NK cells are there, and those patients, so they do have at least some population of T cells, or T-like cells, we should say. The mutation for these patients is most likely in the recombinase genes, which is RAG1 or RAG2. What happens is it'll cause not having functioning RAG1 or RAG2, it'll cause a profound lymphocytopenia. Neither the T cell nor the B cell can rearrange its DNA so that they can produce either the T cell receptor or the immunoglobulin, respectively. So this particular situation is where there are no T's, no B's in the circulating blood is called Omen's syndrome. It was recognized as a syndrome before we identified the particular problem that it was associated with it. They're not going to have HLA expression, right? It'll generally be class 2 expression, right? The one that goes on the immune cells, but you're going to have defects in your HLA expression because part of that is associated with the T cell receptor and how it's expressed on the surface. And you're going to have you're going to have a T cell that, or I'm sorry, a B cell that never produces an immunoglobulin on the surface. This is called bare lymphocyte syndrome. So we had Omen's syndrome and the bare lymphocyte syndrome. Both of these were identified before a possible cause of them was identified. So these are RAG1 or RAG2 and or RAG2 deficiencies. So if the cell cannot produce their immunoglobulin or their T cell receptor, then antigen presentation isn't going to take place. You're going to have a major failure in antigen presentation. No antigen presentation, no immune response. Okay, no adaptive immune response. So these are the major issues that come up with patients with SCID. Okay, these patients are going to usually present pretty early on in infancy, either at birth or over that first year. And it's important that we diagnose these patients pretty early, because if we were to give them, infants, one of the things that we do for the first year is challenge their immune system with vaccines. And some of those vaccines are going to be live. If we were to give a patient with SCID a live vaccine, it could be overwhelming, and it could, depending on which one it is, it could potentially kill them. Okay, so it's important that we try not to give them live vaccines. Okay. Most of them won't, but even, we had the nasal, well, that wasn't approved for infants anyway. But for these patients, our only treatment, they have used enzyme replacement for the ADA deficiency. That's the only one that'll work with. The others, really, it's only bone marrow transplant. You've got to do a bone marrow transplant and get some working immune cells in these patients. Or they probably will succumb to their infections pretty early on. These are the cases that we have bubble boy kind of stuff. These are the cases that you're talking about with those very, very severe infections. Okay. They have found that of the transplants, the cord blood transplants for these patients have worked pretty well. And that's good, because without any treatment for the ADA, we can give them the enzyme. But the other genetic deficiencies, if we don't treat them, these patients usually don't last past the age of 2. Okay, next on the list is Wiskott-Aldrich syndrome. This is it's also a sex-linked syndrome. It is rare. It's only diagnosed about 6 times a year in the United States. That's rare, okay? And it is defined by the triad of symptoms that it causes. And the triad is immunodeficiency, eczema, and thrombocytopenia. You have to see all 3 of those in order to call it Wiskott-Aldrich syndrome. Generally speaking, this has been lethal in childhood because of infection, but also you have thrombocytopenia, so you could have hemorrhage as well. Okay, the other thing that has also occurred is malignancy. So this is a very severe immune deficiency. First things that you're going to see for this one is probably going to be the thrombocytopenia. Patients are going to be bleeding, they're for prolonged periods of time. I mean, we don't do bleeding times directly anymore, but CBC you do on the patient, and they're going to have platelet numbers, through the floor. So um, if we were to do a bone marrow specimen on these patients, they'll actually have either normal to somewhat increased numbers of megakaryocytes. However, that doesn't relate. Those aren't, the maturation does not completely occur. We'd also find abnormalities in their cellular and humoral arms, both arms of the immune system, and the reason is they have defective antigen processing. The deficiency, the way that we see it in the laboratory is their total Ig levels are extremely deficient, right? Severe deficiencies. And that would include their blood group antigens. So the patient, their initial testing, it could be all screwed up because they don't have any natural antibodies. And against the blood group antigens, we usually do expect at least some antibody results for that. So they'll have either very weak or missing antibody reactions, serum reactions in their initial antibody types when they're born. Okay, so these aren't things we ignore. There is a reason why we grade our patient levels. Other things that we might notice in the laboratory is a persistently elevated serum alpha fetoprotein level. And the primary defect seems to be with the CD43 membrane protein. Now that normally is involved in protein glycosylation, which is a step in antigen processing. Pardon me. Interestingly, the gene for this is called WASP, Wiskett-Aldrich syndrome. It is on the X chromosome. You have all kinds of things going on there, wasps, sting, venom, all that stuff, bad thing. So but it is named actually for the guys who named the syndrome, okay? And we know that particular gene where we found the abnormalities will affect signal transduction. So it's not just the CD43 membrane protein directly, but also this WASP gene is affected, and that'll affect signal transduction. Only treatments we have, cord blood transplant from an HLA identical sibling. That's what we got for these patients. And they work. It's just, not an easy process. Okay, next syndrome or deficiency is ataxia telangiectasia. I'm not saying that right. Ataxia telangiectasia. AT, for those of us who are deficient in our enunciations. This is actually also a rare autosomal syndrome, and it's characterized by what's called cerebral ataxia, which is basically a lack of voluntary coordination of muscle movements. And then the other word, telangiectasias, the T part, are the small the small veins become dilated. It's spider veins, okay? And where you would see them in infancy, because obviously we don't expect to see spider veins on an infant, and you would think most of us ladies look to our legs for spider veins, but where you see them on the infants is actually on like their earlobes and in their eyes. So you can see them on various parts of their eyes and in their earlobes. So those are places where you would look for that. These patients also have increased levels of alpha fetoprotein, and the defect here is in a humoral, and the immune portion of problem of this is at both humoral and cellular. Okay, any antibody response, but especially to polysaccharide antigens, is severely limited. Your levels of IgG2, of IgA, and of IgE are often low, if not absent, okay? And the number of circulating T cells is decreased. These patients live a little bit longer. Death will usually occur for these patients in early adulthood, and it'll usually be from some pulmonary disease or some malignancy because they don't have that combined immune response that is required. What's the defect here? Again, there's a lot of research into this, but it seems to be in a gene that's essential for the recombination process for the genes in the immunoglobulin superfamily, okay? That's that chromosome 11 again. There's also a defective kinase, which is involved in DNA repair and in cell cycle control, hence the association with malignancy. Okay, but for our purposes of this lecture, the immune function here, there is a defect on chromosome 11 that prevents recombination of the immunoglobulin superfamily genes. And so you don't get switching, you don't even get it being triggered. No full maturation of these cells. When we look at these, the lymphocytes for patients who have AT, if we do chromosomal studies on them, we frequently see chromosomal breaks and other abnormalities with the T cell receptor genes and the immunoglobulin genes. So the immune portion of it, remember I said that there's a kinase that's deficient, or defective, I should say, a defective kinase involved in DNA repair and in cell cycle control. The immune portion of AT seems to be secondary to that particular kinase issue. Remember, DNA switching, DNA hypermutation, that's supposed to happen in a lymphocyte. But that presumes that you have normal kinases that can help the process along. So DNA switching, that's normal, but you need those kinases there. If they're not there, then that recombination is going to be flawed. From just even the idea of it is going to be flawed. So again, it seems to be secondary to the fact that this kinase really is needed for immune response to occur. Maybe not something that we would normally associate directly with immunology, but all of those DNA repair proteins would obviously be needed. If you're constantly changing around your DNA and removing sections of it, it would be needed. Okay, so if you don't have everything in your toolbox, the whole process is going to be messed up. The only treatment for AT is a bone marrow transplant. And then finally, the reticular dysgenesis. Reticular dysgenesis, another rare genetic disorder of the bone marrow. In this case, the patient has a complete absence of granulocytes and a decreased number of lymphocytes that unfortunately are also abnormal. Because of the absence of those cells, of all those leukocytes, your development of any of your secondary lymphoid organs is going to be pretty poor. If you don't have B cells and T cells to interact inside those lymph nodes, they're not going to be normally formed, okay? Or no germinal centers. They're going to be small, and they're going to be, if you do histology, you're going to be missing all of those germinal centers. This is the most reticular dysgenesis. It's a very severe form of severe combined immunodeficiency. Because it affects all leukocytes, we could say that this is the most severe form of SCID. More severe than the genetic, IL-2RG and all of those, are reticular dysgenesis, because it affects all the lymphocytes together, not just it affects all leukocytes together, not just the lymphocytes. This would be the most severe form, okay? There is an inability in the granulocyte precursors to actually form granules because there's mitochondrial adenylate kinase 2 malfunction, as well as a deficiency in adenylate kinase 2, and that'll cause increased apoptosis of all of the myeloid and lymphoid precursors. Okay, so the patient has some pretty normal clinical things that you're going to find with a patient who has reticular dysgenesis. Some of the first things that are noticed is bilateral sensor neuro deafness, as well as a lack of innate and adaptive immune functions, which usually leads to a fatal, both the innate and the adaptive, remember, they don't have granulocytes, no granulocytes. So the most common problem here is that the patient will develop a fatal septicemia within days after birth if not treated. The only treatment for this is a bone marrow transplant. So unless you know that this is a possibility, not even going to be looking for this, this child is probably going to die of septicemia just within a few days, hence most severe form of SCID. Okay, so you have several types of SCID there, lots of them, but we end with the worst one, of course. And I did try and put it in a little chart for you. I don't know if that helps or not. I liked charts. So just like we can have defects in the red cell line, in the B cell line, in the T cell line, and of course with other things as well, the granulocyte line also, or neutrophils in particular, defects or abnormalities in that particular line can lead to other disorders that we see here. By far, the thing that we rely on most in the neutrophil is the NADPH oxidase system, right? That's the actual system that allows the cell to do the killing. Phagocytizing, frankly, any cell can phagocytize, right? That's actually not a particularly unique thing to the neutrophil, but with the NADPH oxidase system, the destruction of the material that they bring in, that they phagocytize, in particular a live organism, really allows them to be successful in not just phagocytizing, that's just sort of the 10th grade version, right, that you learned what it did. But you know from immunology that the real work is really an antigen presentation, and that requires the NADPH oxidase system. So we're going to see some problems, of course, with that particular, when there are problems with that particular system. So in the chronic granulomatous disease, what we're looking at there's a couple of different kinds. And what we see is that there are X-linked or sex-linked as well as autosomal. More often, these are sex-linked, so more often you're going to see this in men, and it is also more severe. Now remember, I just want to remind you a little bit of your genetics, right? So if you are a woman and you have a flawed gene that leads to a flaw in the NADPH oxidase system, let's say, okay, you still have that second gene that can be expressed, that can give you the healthy enzyme or whatever it is you're missing. That doesn't mean that the flawed enzyme isn't going to be expressed. So you probably have at least some symptoms or some reduction in the system because you're expressing, half the time, you're expressing the flawed gene. Now, in most systems, your body knows that it's flawed and it will overexpress the healthy gene, but that doesn't mean it won't express the flawed gene at all. But in a male, just to stick with the female situation, in the female then, if they have the disease, as long as they're not homozygous recessive for it, they do have it's a little bit more co-dominant than dominant, but they do have a gene that will be expressed that can get them through the process. So they're just expressing less of it, or you could say that they have reduced expression overall of this particular enzyme that they might be missing. Whereas in the male, they don't have another copy, so there's nothing that can help save them. Okay, so if you're thrown into the ocean, as it were, of foreign antigens, you want that life preserver of your immune system. So the woman who's, say, heterozygous for a flawed gene, she might have on, maybe some swimmy arm things. They're not great protection, but they're better than nothing. Whereas the guy's out there by himself, he's got nothing. Okay, so that's kind of the difference, and why you see much more severe disease in the sex-linked variety. There's nothing for them to hold on to on top of that. So what are the symptoms that the patient is going to have? Our big thing when we have flawed neutrophil function, our big thing are these bacterial infections, these really recurrent bacterial infections. So you're talking about, wound infections, pneumonia, osteomyelitis, lymph nodes. They're not just, you normally get a cold or the flu or an infection of any kind, you'll have swollen glands in that area. But in a person who doesn't have properly functioning neutrophils, these are draining adenopathies, right? So they get these huge glands that are draining terribly. They get abscesses. Liver abscesses are a big thing. Dermatitis, skin infections. And what we also see in the laboratory, one of the clues to us is you do a total immunoglobulin level, and it's high, very high. Why? Because again, you've got hyperactivity on the opposite side of where there's hypo. You have some part of the immune system churning away, trying to make up for the other part that's not functioning properly. And that's what we see. We see hypergammaglobulinemia. Okay. Now, the organisms that these patients are infected with, they tend to be of the same group. You get organisms, usually you get a lot of the catalase positive organisms, Staph aureus, you know that one. But then, just to remind you of some of the other ones, you get things that are environmental contaminants. Nobody else would be, have a problem with them, but these patients do. Besopacia, Chromobacterium, fungus. They get fungus. Normal people don't get sick from fungus. These people do because they are immunodeficient. So they get aspergillus. They get nocardia. Okay. So and these start when this is just a child. Before they're a year old, you're like, what the heck? This kid has had so many infections. Okay. And unfortunately, if it's not treated, it's frequently fatal. So just like all these other immune deficiencies, you don't have any protection against the world's going to win. Okay. So CGD, or chronic granulomatous disease, it is a neutrophil dysfunction. You actually there are several things that are going on, but they all essentially result in the inability of the neutrophils to produce any kind of reactive oxygen. That's your NADPH oxidase system. No reactive oxygen means no bacterial killing. That's what we use in our neutrophils to kill. You don't kill it, you don't process. If you don't kill it, you're not going to process it. You don't process it, you're not going to present it. You don't present it, no immune response. So that means you're taking the neutrophils. These are the pawns, right? These are the infantry of your immune response. They're the largest number that you have, and they're what you would consider, unfortunately, disposable. They go out. You can't rely on just dendritic cells to do all the presenting. It just doesn't work that way. The dendritic cells are, they rely on the neutrophils to get the bad stuff to you. There are genes, and they happen to be autosomal, that are involved in all of the subunits of NADPH oxidase. So the NADPH oxidase is the key enzyme that's usually affected here, but it's made up of 3 different subunits. The subunits themselves are actually autosomal, so this would be in the autosomal form. And this is where you can see either, if they're all 3 missing, obviously you have a problem, but usually you have one of the 3 or a number of 3 that are either defective or missing, and you get a badly folded NADP oxidase, okay? So without that oxygen killing, the bacteria are able to to survive because there's no oxidative burst. Okay, remember it's hypochlorous acid that that is normally generated in that oxidative burst, and so without that hypochlorous acid, the bacteria aren't killed. And that hypochlorous acid is also, that oxidative burst, I should say, is also important in the way that we diagnose it. We diagnose it using the nitro-blue tetrazolium staining, or NBT staining. And the reason we can do that is we know that NBT will be reduced by hydrogen peroxide and any other reactive oxygen, and it'll be converted from the NBT, the actual stain is actually colorless, but when you put it in with some kind of reactive oxygen, it'll turn blue. It'll become a blue precipitate. So it's not actually blue when you start, but if you add it, what you're doing, you add the NBT to the tissue section, and if there are neutrophils there, it'll turn blue, and then you can see that. See how wonderful that is? And you can see that with light microscopy. So this was traditionally diagnosed using NBT staining, okay? But with the magic of flow cytometry, we don't need to do that anymore, okay? What we do actually now is they use a labeled dihydrohytamine, which DHR, and that labeled molecule will be reduced in, similarly to the NBT, it'll be reduced in the neutrophils, but it's labeled with a fluorescent label, and so that allows the flow instrument to be able to detect with pretty specific reagent. Okay, so it's dihydrohytamine, and that's used to be able to detect the granulocytes, in particular the neutrophils. And that would be reduced, obviously, in a patient with CGD, okay? Since you won't get, so in the NBT, there'll be no blue precipitate, and on flow, there'll be no fluorescence. Okay, so a lack of fluorescence means the patient most likely has chronic granulomatous disease. Okay, now what's the treatment? Well, we can give you granulocytes, right, because that's one of the components that we have in blood bank. However, that's probably not going to overcome the condition over time. Okay, so that's not going to be sufficient to overcome the condition. They've also had some cytokine treatments, and that has helped some patients as well. But again, over time, it's probably not going to be sufficient to replace the entire immune system. So really the only cure for this is a bone marrow transplant. You really need some cells in there that can produce some functioning neutrophils. So again, we're going to rely on transplant for this to go on. Now we know that if there's a problem with the NADPH oxidase system, you're going to end up with chronic granulomas. You're going to end up with pretty overwhelming disease. But there are some other issues that could also occur. And the next one I have here is the neutrophil glucose-6-phosphate dehydrogenase deficiency. So really long glucose-6-phosphate dehydrogenase, another enzyme, but if you don't produce it, then you're not actually able to produce enough NADPH to feed the NADPH oxidase system. Okay, so you're going to get a similar clinical picture to chronic granulomatous disease. It'll actually, you could call it chronic granulomatous disease, but the enzyme, the cause is different. Picture is going to be the same because you have that whole system not working. Okay, now do we know exactly which genes are tied into this at this point? That's not as clear as with the direct NADPH system. So that one, there's a lot of unknown. We do know that this deficiency exists, but they're still studying it. And again, this is rare. So it's not like this comes up, all the time. We have tons of people we could test this in. This is still pretty rare, and so this is one of those things that has to be figured out sort of into the future. And then finally, another issue is actually a leukocyte adhesion deficiency. So this one, there's actually nothing really wrong with your neutrophils per se. They can oxidatively, they have their oxidative burst, they can kill the organisms. It works out fine that way. However, if they're unable to adhere to CD18, what does CD18 do? Remember back to your immunology. CD18 is a migration molecule, right? So the neutrophils are in your peripheral blood and they move into the tissues in response to an infection. But that moving into the tissues requires that they're able to bind to endothelial markers, CD18, right, and move through the tissues. If they never bind, they're always going to stay in the peripheral blood and never move into the tissues. So you see the problem that's there. So if you get a blood infection, which very few people do, relatively speaking, no problem, you'll be fine. However, if you have a problem like a wound into your tissues, something like that, throat infection, and your neutrophils never respond, then you can see that you're going to have a problem. Both two ways. One, your normal response, which calls for neutrophils to show up, if that's never satisfied, your immune response is going to be, amplified because it's keep calling. What happens when you call somebody, say in your house, like you yell across the room? I do it for my son. I yell for my son, Liam, come. If he doesn't respond, what do? I scream louder. Well, your immune system kind of does the same thing. And eventually you just got people screaming across the house, nothing actually happening. And that's kind of what happens to your immune system. And again, they would work if you could get them there, but they're not there, okay? And so without that, it's actually a particular portion of the CD18 molecule within or a particular part that's missing. And so it won't just be your neutrophils that will be affected. There will be a bunch of other cells too, because CD18 is required for the chemotaxis. It's required for those cells to respond. This is with leukocyte adhesion. It is actually autosomal. That's not, as far as we can tell, sex linked. So, and it's variable. So you don't see an increase in the response. How can these patients will have a different clinical picture, right? So they don't have the same kind of clinical picture. However, they do have an increase. It's not that they have necessarily like crazy number of infections. They will have a little bit more. The biggest problem is the delay in wound healing. The neutrophils are only going to get there by chance. Okay, so you're going to get chronic infections, more especially skin infections, because they're farther away from the neutrophils. You will get intestinal respiratory tract infections that will take longer to heal, and any wound will take longer to heal with this particular picture. So how do we detect this? We can actually see that there's now the antigen for that marker. So you have a CD18 deficiency. How do we detect that? The CD18 is actually on the neutrophil. Okay, it's on the leukocyte. The molecule to which it binds, the LD, which is on the endothelial lining. That's actually the defective part. So what happens is, since it's got nothing to bind to, they're like, well, I'm not going to put this on. If it's not cold outside, you're not putting on a heavy coat, right? I have nothing to worry about. I'm not putting on a heavy coat. And so the cells don't put on their heavy coat. CD is closely tied with CD18 on the leukocytes, closely tied to CD11. So you'll see in your flow reduced CD18 and CD11 expression. And that's how you know that the patient has leukocyte adhesion deficiency. Okay, the complement deficiencies. There's two different categories. Well, first of all, usually we describe the complement deficiencies based on what clinical picture we see. And by doing that, we can divide our deficiencies into two categories. Any disorder of the proteins that inhibit the complement system, like so if you are deficient in the C1 inhibitor, that's going to lead to an overreactive response, right? So the patient is going to have too much bleeding, too much complement activity. So that leads to things like hereditary angioedema. Okay, that's a C1 esterase inhibitor. They could have hemolytic uremic syndrome. That's a factor H inhibitor deficiency. Okay, so that's one classification or category of deficiencies. Now, the second category, do you have a deficiency in a protein that is supposed to activate the complement system, like C3, right? We know that C3 is supposed to activate the complement system. Well, that's going to lead to an underreactive response. So we have the overreactive response. That means you're deficient in an inhibitor. And we have an underactive response. You're deficient in a promoter. So that means you're going to have, now from the immune response, you're going to have a greater susceptibility to infections. Now, there is redundancy in the complement systems as well. So it is likely that there are probably several complement disorders that might never be diagnosed. A patient can probably live if they have either a slight or a mild deficiency in maybe a complement factor that there's a redundancy for. Okay, C1, C3, those ones are pretty important. So a deficiency or weakness in those we would notice pretty quickly. A recent study estimated that at least 10%, that less than 10% of complement deficiencies might be identified. So that means we're missing most of them from that study's perspective. Now, the total hemolytic complement or the CH50 level in the blood, if you have a deficiency, it'll usually be low or undetectable. Those are the things that we're really looking for, and the first one we will look at is a complement level. So that's really our sort of first peek at these kind of deficiencies. But then you can look at the individual ones. So you can measure how much C1 does the patient actually have, and you can actually look at the actual components. Is it C1q? Is it 4? Is it 2? Like which complement is missing? The early components will give the patient sort of a lupus-like syndrome, okay? Deficiencies in the later components, C5 through C9, these are the ones that are associated with an actual infection. And remember, complement is vital in our response against things like Neisseria. So patients who have any deficiency in the later components are much more susceptible to Neisseria infections. Doesn't that sound like a test question? Deficiencies in the later components, 5 through 9, often associated with Neisseria infections. So don't pick C1 as the answer to that one. Okay, give you too much of a hint there. But others, we know, for example, the C1 esterase inhibitor, we know that that's actually a legitimate deficiency, and that this is what we find in patients who have hereditary angioedema. Okay, so that's a distinctive disorder. We know that the patients have that, and we do find a reduction in the C1 esterase inhibitor for that one. All right. And then finally, a nice little chart for you. I like charts, right? So in this one, I give you the screening test that we're going to do. Please look at the first test. What's the first thing we do anytime that we're looking for an immune deficiency? First step is always, always, always going to be do a CBC. Okay, do the CBC with the diff. That's the place we're going to start out. Okay, because if we know right away this patient doesn't produce any leukocytes, well, I think maybe there might be a problem. Okay, so we got to start out with the first step, the bare minimum, before we start getting into all these fancy NBT levels and all that stuff. If you don't have a CBC online, why would you be starting at the end of the road? Okay, so looking at them, look at where we're going to start in the main lab, and you can see CBC with diff, your serum, your total IgG, and then separate the levels. Do I have every isotype out there? The subclasses. Okay, I've got all my IgG, but do I have all the subclasses? We could check that. And then we look at the titers. Do I have, and this would be in the blood bank, how much anti-A am I producing, anti-B, the normal, the natural levels? And then, of course, we can look at specific antibody levels. Do I actually produce IgG levels against specific organisms or specific tests that I can look for. And then we start to look through, if you're suspecting one, can I trigger an immune response? Your immunologist will do this, right? You do it with your allergies. But sometimes they want to do it to see if you'll ever even, generate a response, and they will purposefully challenge you, on your skin, of course, but they'll challenge you with something that's foreign to see if you can generate a response toward it, okay? Your x-rays, of course, to look for the actual structures. Do you have a thymus? If you don't have a thymus, maybe you have DeGeorge syndrome. Okay, so these are the tests that we do. And then the confirmatory tests, some of them we do in the main lab. We can do flow cytometry, right? We can do them in the main lab, or what has become the routine lab. And some of them are a little bit more fancy, right? So the enzyme assays and things like that, those get the chemotaxis assays, those require some, fancy lab work, and those aren't done in the general lab. Much higher level of training for those. But these are the tests that are there. I want you to know them. Like some of them might show up on an exam question. Okay, but that's the whole kit and caboodle for immunodeficiency as far as this class goes.